Hey guys, welcome to the video for chapter 11.2. This video is going to cover all the key terminology from this chapter and hopefully give you a better idea of what's going on. So first off, let's talk about probability. What Gregor Mendel noticed is that the rules of genetics follow the exact same rules as probability. And what that means is this, that every single time your body creates a gamete, there is a 50% chance that that gamete is going to get one allele or another. There's no preference for allele types. If you have a cell that splits into gamete cells, those gametes are going to have a 50-50 chance of having one allele or another. This holds true even with dominant genes. There's a misconception going around in class right now that dominant genes are somehow stronger genes. They're not stronger, they're just programmed differently. So even dominant genes have a 50-50 chance of getting into one gamete or another. And this idea, again, just reiterates this idea, so when I put this onto Edmodo of the notes, you'll at least be able to see it written down. And that's that you have an equal chance of getting one allele or another in a gamete. The next concept that the chapter talks about is something called heterozygous and homozygous. Let's start off with homozygous. A homozygous trait basically means that you have either two dominant alleles or two recessive alleles for a trait. So you're either big T, or little t. The second type is called a heterozygous trait. All this really means is that you have one dominant trait and one recessive trait. Or in the little vernacular here, vernacular is a fancy word, don't worry about it. You get a big T and a little t. Let's give some examples of what this might look like on an exam. So if I ask a question like, if someone is homozygous dominant, what alleles does he have? Well, he's homozygous, that means he falls under this category right here, and he's dominant, which means he has the two bigger of the letters. He has two dominant traits. So you have big T, or whatever you want to choose, big S, big A. All that really means is that he has two dominant traits for an allele. The next question I have here is, if someone is homozygous recessive, what trait does he have? Well, it follows the exact same rule here. Homozygous, okay, he has either two big T's or two little t's. Recessive, well, if you have only two small t's, you are homozygous for a recessive trait, so you would be little t. Okay, so what does this mean for you guys? Well, this goes back to an idea the book talks about next, which is called phenotype versus genotype. This is really easy to understand once you actually understand it, but getting to the point of understanding it is kind of a challenge. Phenotype is basically a fancy way of saying it's what trait you show. So if you are tall, you have a tall phenotype. If you have blue eyes, you have a blue eye phenotype. If you have sickle cell anemia, which is a human disease, you have a sickle cell phenotype. All that's really saying is that it is the trait that you're showing. The other example I have here is if you are blue eyes, your phenotype is blue eyes for eye color. Now, genotype is asking what alleles do you have? So, as you already know, some traits are dominant, like we've talked about with the tall pea plants and the short pea plants. If you have a big T and a little t, even though you have an allele for being short, that little t, you're still going to be tall because you have that gene for big T. What genotype basically asks is, what alleles do you have? And what this whole idea tries to get across is that it's possible to have a recessive allele for something and you may not know it because you have a dominant trait that is overriding, that is basically essentially stronger in some way than the recessive trait. And we'll talk a little bit later about what makes a dominant trait dominant. But for right now, just know that if you have a dominant trait, you're going to have that trait. So here's an example. A person who has sickle cell disease has little s, because sickle cell is a recessive disease. So they have to have two little s's. So their phenotype is sickle cell. Their genotype is little s. Now, if he doesn't have sickle cell, he'll have a big S, or a big S and a small s. Now, in both of these situations here, you don't have sickle cell because sickle cell is recessive. So the phenotype for both of these is no sickle cell. However, the genotype for one of them is big S, and the genotype for the other one is little s. And what this shows you is that the recessive gene can hide the little s, and you may not ever know it. This person right here who has an allele for sickle cell may never know they have one of the sickle cell alleles because it's not something you would see. So a genotype is basically saying what alleles you have, no matter what your phenotype is. Okay, this I wanted to show you really quickly because it's a visual way of seeing it. This was the graph I showed you guys from the last video. But this time, rather than looking over here, I want you guys to look over here. Here we have the genotype, are all big T, little t, for these plants. You have big T, little t, big T, little t. So the genotype for these plants is big T, little t. The phenotype is they're all tall. So even though they have an allele for the little t for being short, they're not actually short because they have the big T to make them tall. Now if we move down to the next generation, what we see is that we actually have three different genotypes. You have a big T, that's one genotype. You have a big T and a small t for these two plants, so that makes a phenotype. And you have, a genotype. And this here is a little t, which is the phenotype for being short. The genotype for being short. Now the phenotype is saying that you have three tall plants and one short plant. So what you get is different ratios. You have, for a genotype, you have a 1 to 2 to 1 ratio. For phenotype, you have a 3 to 1 ratio. I hope that makes sense. And if it doesn't, please feel free to come see me in class. I'll try to find another way to explain it. Okay, let's move on to Punnett squares. Most of you guys know how to do these now. I've seen everybody do at least one, and people are pretty competent about it. What a Punnett square lets you do is that a Punnett square lets you guess the probability of certain alleles. So, what that really is a fancy, because the book's fancy definition for this is a Punnett square lets you figure out the probability of phenotypes and genotypes. Really all it does is it lets you figure out what alleles you have if you were to have these two parents, the mom and the dad, have babies. The graph on page 316 in your book really explains this very well. So if you don't quite understand my explanation, I would highly recommend going to this page in your book, because even though it doesn't have a lot of typing or a lot of words, it shows you visually what's going on. Okay, so here's an example of the Punnett square. If you cross the alleles big A, and little a, what is the probability of having the dominant phenotype? Well, as you learn from segregation, 1A is going to go here and 1A is going to go here, because this is a gamete and this is a gamete. And this can be the mom in this situation. This right here can be the dad, and it'll be a little a and a little a. So what you then do is you then begin to fill in these spaces based upon where letters are at. So you have an A and an A. So one child will have big A, little a. You have a big A and a little a, so the other child will have a big A and little a. And here the same is true. You have a big A, little a, and a big A and little a. So you essentially just draw lines to all the locations and then fill in the appropriate alleles. So to answer the question, what is the probability of having a dominant phenotype? Well, it's 100% because every single child has the possibility of getting a dominant phenotype. And in case I didn't make that clear when I was talking about this earlier, these squares right here all basically represent possible children. Because you have two possibilities, two alleles here and two alleles here for the parents, there's 4 possible combinations of kids. But in this situation, there's no difference. Every single kid is going to be big A, little a, so they'll all have the dominant phenotype. The next kind of Punnett square is the bigger type of Punnett square. This one's a little harder to manage, but you can probably see how it works. What I've done here is that now I have the alleles AA, BB. So this allele, these two alleles here would be for a trait, and these two alleles here would be for a trait. So this could be like blue eyes. This could be, I don't know, let's go with Asperger's syndrome or something like that. Well, as we know, as I'm going to show you right now, alleles separate differently. So you'd have an A and a B. So A and a B would go here. You'd have an A and a B would go here. You'd have a big A, big B would go here. And you'd have a big A and a big B would go here. So all the gametes that this person can produce would be big A, big A, big B, big A, big B, etc., etc. Now for this individual, it would be different. You would have little a, little b, and that would go here. Little a, little b, and that would go here. Little a, little b, and that would go here. And as you probably see, little a, little b, and that would go here. You then do the exact same thing you would do with the last Punnett square, and you go big A, big B, and you put them here, little a, little b, and you get the genotype big A, little a, and big B, little b. Now because there's no difference across any of the alleles here, this is going to hold true for all of them. So if I ask the probability, what is the probability of a recessive phenotype? Well, it's going to be 0 because none of these children here are going to have a little a, or a little b. They're all going to be this combination. So there's no chance of having a recessive phenotype. Now, what this basically teaches us, looking at how alleles separate independently, is this idea of independent assortment. What independent assortment basically means is that one gene does not affect another gene when making gametes. So if you're looking at 2 genes, so you want to have the gene for sickle cell disease and the gene for baldness, for example. Let's have this right here. This will be sickle cell, this will be baldness. What that means is that the genes for sickle cell right here do not affect the genes for baldness. They're independent. They sort independently. And this just puts home the idea. The A alleles will not affect the B alleles. I hope that answers most of the major questions for you guys. I'm hoping to do some videos on Punnett squares in the future. I'm trying to find a better way to do it. PowerPoint is terrible for demonstrating Punnett squares because really it's better to have some kind of an animation. So I'm trying to get some way to show you guys this content without having to use PowerPoint. So I don't know when I'll get these videos up, but I will try to get something up before you guys have the exam so you can see plenty of examples of how to run Punnett squares. Worst case scenario, I'll find somebody else's videos online you guys can use, and hopefully that'll help you guys understand the content. And that's pretty much it. Thanks for watching. I hope this helps you guys out, and I will see you guys in class.